Hello students, I am Ms. Grishma Dalal. Today we are going to study in pharmaceutical biotechnology, enzyme biotechnology. This is lecture 2 in which you are going to study in detail about enzyme biotechnology. What are the methods of enzyme immobilization and their applications. So let us start with basic, like this is the introduction part. Here, enzymes. First of all, you should know what enzymes are. Enzymes are natural proteins produced in tiny quantities by all living organisms and functioning as highly selective biochemical catalyst in converting one molecule into another. So now, I will explain you slowly. Enzymes are the natural proteins that are produced in tiny quantities by all the living organisms, which is natural, right? And functioning as highly selective biochemical. They are very selective in nature and they are biochemical catalyst. What do you mean by catalyst? Catalyst is something that will boost your reaction, right, in converting one molecule into another, right? So when one molecule gets converted into another, you will need some sort of catalyst is nothing but it will boost the conversion, right? Speed up metabolic reactions to a very great extent but do not undergo any change in themselves. This is really important to remember, that it will speed up the reactions, metabolic reactions, to a very great extent, but they will not undergo any change themselves. So now when we talk about the enzymes, you should know some of the general properties. So we will take a quick look. The catalytic properties of enzymes are responsible to speed up chemical reactions but remain undestroyed at the end of the reaction. As mentioned in the definition itself, that the catalytic properties of enzyme are responsible only to speed up the chemical reaction, but it will remain undestroyed at the end of the reaction. Second, it works in either direction. It catalyzes the forward and backward reaction to the same extent. The direction in which the reaction goes will depend on the relative amount of substrate and the product present. So amount is really important. We will come across the words substrate and product in the coming video. Number 3 is changes the rate only at which chemical equilibrium is reached. It does not affect the position of the equilibrium. It will only change the rate. This is a must to keep in mind, that it will only change the rate at which the chemical equilibrium is reached or you will acquire. But it will not affect the position of the equilibrium. It will not change the equilibrium for all, right? Only the slight rate will be changed. The next property is an enzyme speeds up the rate of reaction by lowering the activation energy. It will speed up the reaction, as mentioned again in the definition, by lowering its activation energy. We will come across through it. It works rapidly and therefore required in small quantity. It is soluble in water and works in aqueous solution in living cells. All enzymes operate only on specific substrate. Only substrate of particular shape will fit the active site of an enzyme. Please keep this point in mind because we are going to study in detail about this point. I repeat, all enzymes operate only on specific substrates. Only substrates of particular shape will fit the active site of an enzyme. All enzymes are proteins. You need to keep this in mind. What? All enzymes are proteins. The activity of an enzyme is affected by temperature. Optimum temperature gives maximum activity. Please keep this point in mind because it will be really helpful in your coming topic. I repeat, the activity of an enzyme is affected by temperature. Temperature is really important when it comes to an enzyme, right? And Also, the eighth point is connected, so keep that in mind. So 7, 8 and ninth. These three properties are extremely important properties and you should not neglect these properties even while writing your theory exam. So, I repeat all the points again because it will be helpful and it will be connected in your coming video topics. Number 7. All enzymes operate only on specific substrate. Only substrate of particular shape will fit the active site of an enzyme. Number 8. All enzymes are proteins. And number 9. The activity of an enzyme is affected by temperature. Optimum temperature gives the maximum activity. Number 10. Enzyme activity is affected by pH of the medium. It works best at an optimum pH. This is also again one of the most important property that you have to keep in mind and do not neglect this point. Enzyme activity is affected by pH of the medium. It works best at optimum pH. Number 11. Enzyme may be denatured by excessive heat, extreme pH or various chemicals. This is because enzymes are soluble proteins. Again, this point is important. I repeat, enzyme may be denatured by excessive heat. This is when you apply excessive heat, they cannot remain stable and they get denatured. Extreme pH is also responsible and sometimes various chemicals are also responsible for denaturation. Some enzymes work efficiently only in the presence of appropriate cofactors. There are several cofactors. Now you will be surprised to know what cofactor and also curious to know what the cofactors are. Cofactor is nothing but it is a non-protein substance which is essential for some enzyme to function efficiently. Which means when I said to remember the last point, enzymes are proteins, right? So these cofactors are a non-protein substance that will help some enzyme to work or function efficiently. Basically, there are three types: activators, coenzymes, and prosthetic groups. Activators are substances which are necessary for functioning of certain enzymes. Coenzymes are non-protein organic substances which are essential to efficient functioning of some enzyme but are not themselves bound to enzymes. And prosthetic groups are organic non-protein molecules and they are bound to enzymes. So keep in mind these three: activators, enzymes, coenzymes and prosthetic groups. These are three types of cofactors. Here they are. Cofactors include activators, coenzymes and prosthetic groups. So now we are coming really close to what is important. Here is one of them, that is nomenclature and classification of enzymes. IUB, that is International Union of Biochemistry. Please note it down. International Union of Biochemistry initiated standards of enzyme nomenclature and each enzyme has unique name and code number that reflects the type of reaction catalyzed and the substrate involved. Which means they have kept a code number that will reflect the type of reaction that occurred or that is catalyzed and which substrate is involved. The EC number having 4 digits to categorize the enzyme. EC number again is enzyme commission number having 4 digits to categorize. So here are the 4 digit. What are the 4 digit? The first digit in every EC number is the major class. Right. It will be the first number will indicate the major class to which the enzyme belongs. Right. The second number is the type of group that is involved. The third group is the reaction more precisely indicating substrate on which group acts. And the fourth number and the last but not the least is serial number of the enzyme. So you need to remember this: first digit, second digit, third digit. It is just like IUPAC classification, right? Enzymes are having a unique name and code number, right? So this enzyme commission number will have 4 digits. The first digit is the major class to which the enzyme will belong. Second digit is type of group that is involved, and third group is the reaction more precisely indicating substrate on which the group acts, and the fourth is serial number of the enzyme. Here is the example. These are some of the example of major classes of enzymes. Have a look at it. You can pause the video here and write it down. Now if we see class, type of reaction catalyzed, and example. So in class EC1 oxidoreductase, EC2 transferase, EC3 hydrolase, EC4 lyase, EC5 isomerase, EC6 is ligase. I repeat, EC is the full form of enzyme commission number, that is EC1 oxidoreductase. If I want to make you understand, then there is one example, that is EC.2.7.11, which represents that EC, that is enzyme commission 2, class 2 belongs to transferase, as you can see here, right? In second row it is transferase, right? Subclass is 7, that is transfer of phosphate, which is not shown here, right? Subclass 1, an alcohol group as phosphate acceptor, and the final digit 1 denotes the serial number of enzyme in transferase class, that is hexokinase, first isolated enzyme in transferase category. So in here you need not to remember everything, just you need to have the knowledge of which are the major classes of enzyme. Like for class 1 oxidoreductase, the example will be lactate dehydrogenase and oxidase, right? Or it can be more than that also. But what is the main role of oxidoreductase? So in the center, as you can see, the center column, type of reaction catalyzed. So what will oxidoreductase will be responsible is to catalyze oxidation reduction reactions, right? Or they can also help in transfer of H and O, atoms of electrons from one substance to another, right? The similar way, if we go on further, EC2, that is class 2 transferase, they will be responsible for the reaction of transfer of functional group from one substance to another. Example, transaminase kinase. Now these examples are what? These examples are examples of enzymes, right? So don't confuse. You can call it proteins because enzymes are ultimately proteins, right? Same goes for EC3, EC4, EC5 and EC6. Hydrolase will be responsible for formation of two products from a substrate by hydrolysis. Don't forget that these type of reaction will be catalyzed, right? Because enzymes are acting as a catalyst, as a biochemical catalyst that will boost the reaction. So you need to understand that this will boost formation of two products from substrate by hydrolysis and so on. This belongs to every class. This belongs to every example that is mentioned in this table, right? So again, I will come back to the point, that is hydrolase. The examples are lipase, amylase, peptidase and phosphatase. EC4 lyase, non-hydrolytic addition or removal of group of substrate, decarboxylase, etc. Isomerase will be responsible for intramolecular rearrangement. Isomerase and mutase are the example. And ligases will join two molecules by synthesis of new CO, CS, CC bond with simultaneous breakdown of ATP. And the example is synthetase. Your homework here is to know the full form of ATP, right? I know we will see it in our further syllabus and chapter, but for now, for whoever is watching this video, will have to know and search what the full form of ATP belongs. Now coming to the main point, as I said, this mechanism of enzyme action. So basic mechanism of enzyme action, if we see, it is when a substrate binds to the active site on the enzyme. This is the basic mechanism you need to get into your mind very clearly to understand this. The basic mechanism of enzyme action is binding of substrate to active site on the enzyme. Now, active site. What is active site? It is a specific region of enzyme which combines the substrate. It will combine all the substrate. Like it is just like how in college, right? If you are in college, your college is surrounding wall and you are inside it, right? So it is just like that. It is a specific region of enzyme which combines the substrate. This leads to changes in the distribution of electrons in the chemical bonds of the substrate, which causes the reactions that lead to the formation of the product. So what happens after the active site, and you know the basic mechanism now. So what will happen next? This will lead to the change in distribution of electron in chemical bond of the substrate, which will cause the reaction that will lead to formation of the product. It is like A plus B will give you AB, right? It is just like that. Products are released from the enzyme surface to regenerate the enzyme for another reaction cycle. Now, products are released from the enzyme surface to regenerate the enzyme. Don't worry, you will understand this once we get into the theory. So why not go fast? Two theories. First, you need to understand this, right? E plus S will give P plus E, right? Isn't it simple? E is nothing but the enzyme, S is nothing but the substrate. When they both combine or they both react, you will get P, which will be product, right? And this is E and P. This is enzyme product complex, right? To understand this whole mechanism, there are two theories, major theories that are really important. First is lock and key hypothesis, enzyme plus substrate. Obviously, first enzyme plus substrate, they both will combine. This theory was first postulated by Emil Fischer. Remember this. This will be really helpful in your competitive exams. So log is basically enzyme and key is basically a substrate. You need to remember this. It is really fun to remember this model because here you can imagine that you are locking something, or at your home you are using the lock and key, right? It is just like that way. Only correct size key substrate will fit into the key, that is active site of the lock enzyme. So you need to remember only two things, that lock is equal to enzyme and key is equal to substrate, right? Now only the correct size key will fit into the key, that is active site of the lock. It means the lock is the enzyme, right? So the key must be right. Now lock and key theory is just like this. As you can see, there is a substrate which is blue color, enzyme is yellow color, and active site where it will combine, right? So it will give you when they both combine, you can see the structure, right? It is similar, it is correct. That's why it will combine and it will give enzyme substrate complex, which in short it is also called ES complex in many books or in internet sites. This is the lock and key model. The substrate and enzyme active site have complementary shapes, as you can see, right? Now video time. Learn the videos in a really fun and interesting way. If you want to understand this really fun concept of mechanism of enzyme. You need to watch the videos that are linked in the description for clearing the concept well. Just switch on the subtitles, watch the video. You can pause the video here and watch that those videos come, or you can just remember the time where you stop this video, and you can watch that video and come back again and continue from here. But please watch those videos that are linked in the description. They will be really and extremely beneficial for your knowledge. So now I'm continuing by the theory. I hope you guys have watched those videos. That will be really extremely helpful in your further study. So let us go further. Induced fit model. As you can see in description, there are two videos that I have linked. First one was lock and key, and the second one was induced fit model. Now I hope that something at least is clear in your mind as to what induced fit model is. So let's see. Proposed by Koshland. Again, remember the scientist name. It will be helpful in your competitive exams. In the presence of substrate, active site of the enzyme may change the shape to fit the substrate. That is, the enzyme is flexible and molds to fit the substrate molecule. Right, so it will be really helpful. This theory is stated based on the nature of enzyme, that is protein molecule is flexible enough to allow conformational changes. So it is all dependent on what in lock and key it was not flexible, but in induced fit model it is conformational changes. It allows conformational, which means that it is flexible enough to change the shape. Now induced fit theory is this way. You can pause the video here. In this induced fit theory, as you can see, the substrate are of colored green. At first they are green color. As soon as they combine to enzyme substrate, enzyme plus substrate, when they enter the active site and they combine with the enzyme, they form a complex. That is the second figure, enzyme substrate complex. Then again, as soon as they combine, they will start getting converted into the product, so it will form as enzyme product complex. And then last but not the least, you will get enzyme will be removed and the product will be removed aside, leaving the active site. So it is really amazing. At first you can see the active site doesn't have perfect shape. They will get changed into they will fluctuate their side, and it will try to make the shape of the substrate and active site both feasible, right, so that they can fit inside them. So this was all about induced fit model. Now we are going to study about factors affect the enzyme activity. First one is concentration of the enzyme. Concentration of the enzyme is increased, the velocity of the reaction proportionately increases. You need to understand this by using this graph. You can see it. As I mentioned, there are factors that affect. We have already seen that two factors are most important, that is temperature and pH. We are going to study about that. But the first important factor is concentration of enzyme is that much important. Concentration of enzyme is when you increase the concentration, the velocity will increase proportionately. Right. As you can see in the graph, it is written that you can see the straight line, right? So that it is increasing. As you increase the enzyme concentration, the graph is also increasing. What does this indicate? That velocity of the reaction depends on the enzyme concentration, right? The second one is concentration of the substrate. Increase in the substrate concentration gradually increases the velocity of enzyme reaction within the limited range of substrate level. I repeat, increase in substrate concentration gradually increases the velocity of enzyme reaction within limited range of substrate level. A rectangular hyperbola is obtained when the velocity is plotted against the substrate concentration, which is shown in this graph. So now what is this? In this it is shown that substrate concentration, when you increase the substrate concentration, the velocity will also increase, but it will be in a limited range, right? And that's why you can see, you can get a rectangular hyperbola type of plot, right? You can see, and there are also what can we say, there are, you can see the phases A, B and C. And the one new thing that in this graph is this Km. So let us see what Km is. This is about enzyme kinetics and Km value. As you can see, uh, the enzyme is E and substrate is S. And when these both combine with each other, they form an unstable enzyme substrate complex ES for the formation of the product P, right? So this is all about the first equation. In this equation you can see that there are K1, K2, K3, right? So here basically K2 is used, K1 is used twice, right? So basically these are velocity constant for respective reactions. Now, the second equation here, this Km is equal to K2 plus K3 divided by K1, the Michaelis-Menten constant. Km is nothing but what? Michaelis-Menten constant. Or you can also call, it is also known as Briggs and Haldane's constant, but most widely it is called Michaelis or Michaelis-Menten constant. Whatever you can pronounce it, is given by the formula. And again, the third and the last formula is what? About after adding a suitable algebraic manipulation, you will get V is equal to V max into the bracket S divided by Km plus into the bracket S. Right. Now, to understand what are the alphabets, so first of all, V is measured velocity. If we talk about V max, it is maximum velocity. If we talk about S, it is what? Substrate concentration. And Km is nothing but Michaelis Menten constant. Remember this. This is the way. This is extremely important equations, and this is the most important topic. This is what are we studying? This is all about concentration of the substrate. Now, how it is connected? Km or Michaelis-Menten constant is defined as the substrate concentration to produce half maximum velocity in an enzyme catalyzed reaction. It indicates that half of the enzyme molecules are bound with the substrate molecule when the substrate concentration equals to Km value. So, Michaelis-Menten constant, as you can see, it is the substrate concentration to produce half maximum. It is nothing but it is a substrate concentration to produce half maximum velocity, not full, in an enzyme catalyzed reaction. It indicates half of the enzyme molecule are bound with the substrate already when the substrate concentration is equal to Km value. Km value is a constant and a characteristic feature of a given enzyme. It is representative for measuring strength of ES complex. Now, what does this mean? When you get low Km value, it indicates that there are strong affinity between enzyme and substrate. When you get high Km value, it will reflect that there is a weak. Sorry for the spelling mistake. It will reflect that weak affinity between them. It is WEAK. Sorry for the mistake again. For majority of enzymes, the Km values are in the range of 10 raised to the power minus 5 to 10 raised to the power minus 2 moles. Line-Weber-Burke double reciprocal plot. So this is the next plot that we are going to study in this. For the determination of again Km value, the substrate saturation curve is not very accurate since Vmax is approached asymptomatically. So now here in Line-Weber-Burke double reciprocal plot, you need to understand that for this is for the determination of Km value. If you want to derive a Km value, the substrate saturation curve is not very accurate because Vmax will be, you cannot control the Vmax, right? It will not be accurate. Vmax is nothing but maximum velocity that is obtained once the reaction is start, right? So for the determination of Km value, we are using Line-Weaver-Burke double reciprocal plot. By taking reciprocals of equation 1, a straight line graphic representation is obtained. Here in equation 1 is the first equation that I showed you in enzyme kinetics and Km value. You can go back to the video where enzyme kinetics was the heading, and you can see that the first equation E plus S, which first makes a complex ES, and then it gives us a product E plus P, right? Now, if you consider that equation, you will get a straight line graphic representation. The Line-Weaver-Burke plot is shown. It is much easier to calculate Km from intercept on x-axis, which is minus 1 divided by Km. Further, double reciprocal plot is useful in understanding effect of various inhibition. So this is like this way. You can see in this graph, it is what it is. Why it is called double reciprocal plot? Because you can see that there are partitions here, right? And it is double reciprocal plot. It is just like how graphs you can divide into positive and into negative, right? So this on the x-axis you will see that it is minus 1 divided by Km, right? So this is the only reason why it is called double reciprocal plot, and it is really helpful in understanding the effect of various inhibitions. You can see 1 by V, 1 by V max, right, at the point that is plotted here, and here you can also calculate the slope, right? Here, this slope is equal to Km divided by V max. So this is again extremely helpful, and it will help you in understanding what the Km value you will get, and by that Km value you will understand the affinity between the enzyme and substrate molecule. Number 3 is effect of temperature, the extremely important point. Velocity of an enzyme reaction increases with increase in temperature up to a max and then declines. This is extremely important, please remember, right? A bell-shaped curve is usually observed. This is different from the other factors that we have seen. The graph is totally different, so you need to keep this in mind, right? The optimum temperature for most enzyme is 40 degree to 45 degrees Celsius. Extremely important point. Keep this in mind, and this can also be asked in your competitive exams, so keep in mind, right? In general, when the enzymes are exposed to a temperature above 50 degrees Celsius, denaturation leading to rearrangement in the native or tertiary structure of the protein and active site are seen. So in general, when the enzymes are exposed, you expose some enzyme to a higher temperature which are more than 50 degree, automatically the enzymes start to get denatured, leading to rearrangement in the tertiary structure of the protein and active site are seen. Majority of the enzymes become inactive at higher temperature. Extremely important. Remember this point. So now see really carefully. You can pause the video here, understand the graph. Okay. In this graph, as you can see, the temperature and enzyme velocity both are connected, right? When you increase the temperature, and as you can see, if you increase the temperature, the graph drops, right? So you can understand that why they have they are so much sensitive, because it is a fact that enzymes, which are generally also sometimes called as protein, they all get denatured at higher temperature. Remember this graph. Effect of pH. Increase in hydrogen ion concentration, pH, considerably influences the enzyme activity, and a bell-shaped curve is normally obtained. This is the same case as of temperature, but don't mix the graph, right? Each enzyme has an optimum pH at which velocity is maximum, just like the temperature. Most of the enzymes of higher organisms show optimum activity around neutral pH, that is 6 to 8. Again, remember this is extremely important, and it will be helpful in your competitive exams too. There are, however, many exceptions like pepsin, that pH is usually 1 to 2. This pepsin enzyme, and then acid phosphatase 10 to 11 for optimum. These are what? These are some exceptional case. which will not have any effect of pH, right? So this is the graph. It is really similar to the temperature graph. As you can see that pH is written and enzyme velocity is written. Now you can see the bell shaped here too. But as you increase the pH, except for exceptional cases, the all enzymes will definitely get denatured. Now, if we see, we compare these two, right? First is temperature, the second one is pH. So, again, I am speaking, don't draw the graphs correctly because temperature is different. You can see on the axis that it is 10 degree, 20 degree, 30 degree, 40 degree, right? So, don't mix up this. These both are almost close, and remember this factors. These are really extremely important. And last but not the least is effect of product concentration. So the the accumulation of reaction products generally decreases the enzyme velocity. For certain enzymes, the products combine with the active site of enzyme and form a loose complex and thus inhibit the enzyme activity. So sometimes when accumulation of reaction product occurs, it decreases the enzyme velocity. So for certain enzymes, the product combines with active site of enzyme and form a loose complex, and thus they inhibit the enzyme activity. They stop the enzyme activity. So it's better not to accumulate the reaction products. Right? Now, in the living system, this type of inhibition is generally prevented by a quick removal of products formed. So, you need to remove quickly as possible. Just understand this factor that accumulation is not good for enzyme or any reaction that occurs with the help of the enzyme. So, accumulation or accumulated products should be immediately be removed. In living system, there is a natural uh, what can we say? It is a natural thing that it removes the products that are formed, so that the enzyme velocity is maintained and the process is good. So this was all about enzyme, and in today's lecture we have studied everything about basic thing about what is enzyme and what happens when it combines with the substrate, and what happens when it is inside the body of higher organisms, and we have studied some of the factors and some of the theories. This video is extremely important as of for the exam point of view, and make sure to watch the videos that are in the description to understand more well about the enzyme theories. That will clear your mind, and don't skip anything, otherwise you will be in a great loss. So thank you so much, and I will continue my next session with the new topic, that is immobilization of enzymes. So till then, keep this video safe, and you can study well through this topic, and just wait for the lecture 3, that is immobilization of the enzymes. Thank you.